Well, good morning. It's really my great honor to introduce the Slatopolsky lectureship today. Dr. Slatopolsky truly embodied the triple threat with his remarkable contributions in patient care, teaching, and scientific investigation. He trained generations of academic nephrologists and scientists that carry on his legacy today in their own research laboratories. With his own groundbreaking work in the areas of parathyroid hormone, vitamin D biology, chronic kidney disease, and metabolic bone disease, Dr. Slatopolsky's discoveries improved the lives of our patients who suffer from kidney disease. In this renal division photo from 1981, we can see Dr. Slatopolsky right there next to Salo Klar. There's Mabel Perkerson, Mark Hammerman, my predecessor, and other members of the division. And it's hard to believe that, but Eduardo had already been in the division for 15 years when this photo was taken. This is Dr. Slatopolsky's famous rooster called Macho. He was mean, but produced the most potent and specific antibody against PTH the world had seen and enabled countless laboratories to measure PTH levels for the first time. We still have Macho in the renal division today. I'm not sure, we might be breaking some rules with that, but anyway. It's not an exaggeration to say that we in nephrology viewed Eduardo as Superman, but really also appreciated his wonderful sense of humor and panache. We were humbled and grateful at Ed's remarkable generosity to the division and school as well. He established the Eduardo and Judith Slatopolsky Professorship in Medicine and Nephrology, and in 2022 we were able to celebrate Jeff Miner as the inaugural incumbent. Dr. Slatopolsky passed away this past April here in Barnes, and having attended renal grand rounds in our division just the week before. We will be holding a celebration of Ed's life on December 12th, which would have been his 90th birthday. It is truly a pleasure to introduce and welcome back our speaker this morning, Dr. Toby Huber. Toby received his medical degree from the University of Freiburg, followed by clinical training at the University Medical Center, Freiburg. And from 2003 to 2006, Toby and his family lived here in St. Louis, where he pursued postdoctoral training in the laboratory of Andre Shaw in the Department of Pathology and Immunology. And I'm thrilled to welcome back Andre here in our audience today as Toby's mentor, having made the trip out from San Francisco, where Andre runs a research group in Genentech for the last 9 years. So this is quite a homecoming. Toby has made numerous fundamental contributions to our understanding of glomerular disease across the entire spectrum, from basic mechanisms to clinical trials. He has received too many honors to list, but including ASCI, AAP. He was the Donald Selden Young Investigator Awardee by the ASN, and he's one of the most cited researchers in all of nephrology. He is currently chairman of the Center for Internal Medicine at the University Medical Center Hamburg, where he is founding director of the Hamburg Center for Kidney Health and the inaugural president of the International Society of Glomerular Disease. We are very proud to call Toby an alumnus of WashU, part of our family, and we're very grateful to him for delivering this year's Slatopolsky lecture entitled The Future of Kidney Health, From Disease Endotyping to Clinical Translation. Please join me in welcoming Toby.
Dear Ben, thank you very much for the introduction. Dear friends, dear colleagues, dear WashU family, that's what we are calling it. And not just calling it, but truly feeling it by heart. Dear Andre, as my mentor, I'm very happy that you're here. Dear Jeff, as one of my mentors at the time. I would just to briefly remind on Eduardo, because he bridged as very few people the past, the present, and the future. The past, because he was going back to Neil Bricker and telling all the old stories. The present at the time, because he was so sharp and interested in all ongoing experiments and doing experiments himself. And the future, because the way how he portrayed complex organ physiology, how he brought it to complex animal models and then translating it back to humans, has never been more actual than today, with all the data that we're receiving from highly resolved single cell and other highly resolved spatial techniques. This complex organ physiology that he really pioneered is becoming more important than ever before. These are my disclosures, the exchange that we have all the time, and everybody. This is just the faculty that I could meet over 2 days. And of course, I applaud this division for being front-running, not just in this country, but internationally and globally, with so many things and activities going on here. Now, I know there are also, lots of residents here, so not everybody is a nephrologist, and not everybody might be aware of what we call the kidney health crisis. So it's almost 10% of the world's population that is being affected by chronic kidney disease. And chronic kidney diseases are rising as a reason for mortality. As you can see here, it's predicted in 2040 to be one of the key and main mortality factors. And there was a recent editorial in Nature pointing out that CKD is the quiet epidemic at the time, but there are still significant data gaps, and we need to prioritize kidney research and collaborative research in the area of kidney, but also inter-organ signaling. Now, going one step further to understand the largest knowledge gaps that we have in the field. It's interesting, remarkable, this very recent data being published from a UK biobank showing that rare kidney diseases, just accounting for 5 to 8% of all kidney diseases, make up more than 30% of end-stage renal disease and kidney failure. So it's really the rare kidney diseases where we can't offer much to slow down progression, to hold or to reverse. And it's also being portrayed here very nicely that these rare kidney diseases are the ones showing the quickest and fastest progression in renal functional decline. And most of them are actually driven by the immune system. Most of them, some of them are genetically driven, most of them are immune driven. And many of them are immune driven glomerular diseases. So this is the space where we need to fill the knowledge gaps. And why is this? Why don't we have better solutions as of today for inflammatory glomerular kidney diseases? And it's still rooting in the ill definition of these diseases. We are still using definitions of these kidney diseases, and it might sound incredible to a cancer or cardiologist or, internists from other fields, but we are still using a 50-year-old nomenclature, which is just the histotype. So we are saying membranous nephropathy if you see thickened membrane, minimal change if you don't see much changes by histology, or crescentic if you see crescents. This is the level still of today how we describe our diseases. And by that, we prevent to include just in the nomenclature the pathophysiological information, the activity of a disease, and we prevent precise and individualized drug selection and improved outcome predictions. So kidney immunology is certainly very important. That's why I need to remind that one of the pioneers in kidney immunobiology was Emil Unanue. He was the first one to use nephrotoxic serum in the 60s to study the immune system portrayed at the kidney using the system, and at his time at La Jolla at the Scripps Institute, and then bringing it to St. Louis and being the mentor of Andre Shaw. And that's why we kept talking about Emil in our lab so much. And he was showing us, how to use nephrotoxic serum to induce an aggressive inflammatory GN. And then it was really kind of in the next generation, the work of Andre Shaw's lab, also together with Jeff Miner, that brought back the idea looking at the immune system and kidney at the same time, understanding even overlapping functions of molecules in both systems, and trying to include immunobiology thinking in how we view the kidney and how we try to understand diseases. And these two papers from Andre and from Andre and Jeff. These were the ones that basically prompted me or made my decision to want to join WashU and Andre's lab. I should also mention, because John Atkinson is here, the pioneer of complement, and all your work and your groundwork that you were laying out is now coming full action to the kidney. We are just coming back from the ASN Kidney Week in San Diego. Half of the talks were focusing on complement and the role of complement in the kidney. So another thing going back to WashU. So how are we going to solve this problem of these ill-defined diseases, and how can we reconceptualize how we approach kidney diseases? And that's a concept I would really like to introduce at WashU, because this is the best place to introduce new concepts. So I would like to suggest that we rethink kidney diseases and how we are going to do this. So we used to use the factors that drive a disease, like hypertension or diabetes. We use them as terms to describe a disease: diabetic nephropathy, hypertensive nephropathy. But these are not the kidney diseases. I would rather like to call them the disease factors, because they have a variable penetrance. Sometimes they lead to kidney disease, sometimes they don't. So let's think of these things like the genetic architecture, the hemodynamic, metabolic, inflammatory, environmental factors as the CKD disease factors. But they are not the kidney disease themselves. The kidney disease is being originated by the response of the kidney tissue towards the CKD factors, right? So now we have the CKD disease factors, and the way how the kidney responds will determine whether we develop and to what extent we develop kidney diseases. So for that, the kidney disease, which we would like to be calling endotype, which is the endotype is the pathobiological driver, the checkpoints of disease progression. This is the tissue response, and then we can basically break down the tissue response at the cellular level of immune cells, epithelial cells, interstitial cells, in the interaction between the cellular compartments, and also how these compartments are changing, or we call it phase transition from to fibrosis eventually, and stages which cannot be reverted. And then we also always keep thinking on the organismal impact, because we know that the kidney is interacting with all organs, and CKD sends signal to all other organs. So kidney disease is never being an isolated disease. It's always affecting organismal health. So this is the endotype concept. Now, if we believe in this endotype concept, this should be the groundwork to redefine how we diagnose kidney diseases. And I would like to suggest a kind of DCT classification, similar to people are doing it in the oncology field. So by describing the disease factors, be it immune, be it diabetic, metabolic, genetic, then the eventually circulating factors like autoantibodies, and then the tissue response. And by that, if we would follow this path, then we would name, and I will give one example today, then we would name the disease factors or circulating factors, which eventually can be used as biomarkers, and the tissue response. And by that, we classify and stratify diseases along their pathobiology or the endotype. And this then, of course, easily translates into new therapeutic interventions, because if we understand the diseases better, what they truly are, we can refine existing therapies. We can use complement inhibitors in various diseases, for example. We can repurpose existing therapies like B-cell targeting and others. And of course, we can also strategically look for novel interventions and directly aiming for some of key checkpoints and drivers of disease. Now, at the end, because this is a community thing, we would like to integrate all data in some kind of virtual kidney project that at a certain time point will allow us to manipulate and predictively manipulate pathways in silico and predict their outcome, and by that, refine and repurpose drugs even quicker. And and if you think about the stream of this human virtual kidney project, then again things come back to WashU, which has been a key part also of a genome sequencing project over 25 years ago, and And very recently, the landmark paper on the human cell atlas came out with, and it was coordinated by Sanjay. This has really become, and the paper was published last year, it became now a major, Sanjay became the major resource of the community, not just in the United States, but across the globe, to annotate our diseases and our single cell profiling towards this atlas. So again, something that has been importantly been done. And I would also like to mention Ben's work. Ben has become the global leader in highly resolved single-cell data and spatially resolved data. There is no other laboratory, and I was really amazed when I looked it up, but you published over 60 papers since you joined WashU on single-cell resolved information, which has really become the landmark and the leading force in this key area of nephrology. So congratulations to you, your division, and all your team doing all this work. This is really absolutely remarkable. Now, let me give one example how we understand how this endotype-based restratification of kidney diseases could work on one recent example from our team. And it goes back to a term which we shouldn't even use anymore. Idiopathic nephrotic syndrome means you talk to your patient and say, you have a disease which I don't know where it's coming from. That's what idiopathic means. Idiopathic means, we don't know what's going on. And we really also in this place, let's jointly try to remove these terms idiopathic. I think they're out of time. We don't want to see them anymore. We don't want to teach our medical students about idiopathic diseases. So idiopathic nephrotic syndrome is massive proteinuria, massive hyperalbuminuria, massive swelling and edema, high risk for infections and thromboembolism, so it's a life-threatening condition. Now, the history of this idiopathic nephrotic syndrome goes back over 2,000 years. So Hippocrates described it first, 400 BC. He was so cleverly and well observing. So he observed that the urine indicates these patients when there are massive bubbles, the protein in the urine causing this foam and bubbles. So that's what he observed, and that's how he called the disease, 400 BC. It took almost 2,000 years until Cornelius Reulands of Belgium, one of the first, we can maybe eventually even call him the first pediatrician, described it in a little bit more detail, particularly the edema and the massive swelling, and he suggested wrappings to help with the symptoms. And then another 500 years, the first treatment was suggested in 1952. Believe it or not, this 1952 treatment is still our first choice treatment in idiopathic nephrotic syndrome. So, we haven't been moving very quickly in the last 2,500 years. So how can we accelerate discovery paths? So a key finding that helped understanding leakage of the filtration barrier came with the identification of nephrin by Carl Tryggvason's group 25 years ago. And then shortly after, teams of David Selland and others could show that antibodies directed against this novel transmembrane protein, forming obviously the kidney filtration barrier, are inducing rapid onset massive proteinuria. And then these antibodies have been observed in people being transplanted with Finn-Meyer mutations, and some of them suffered from a quick recurrence, and there also anti-nephrin antibodies could be detected. Some people even claimed that these antibodies might play a role in type 1 diabetes, which never have been independently proven. And then 2 years ago, it was a Boston group that was suggesting anti-nephrin antibodies in 18 cases of minimal change disease. So something was going on with these understanding the kidney filtration barrier build up of nephrin, but also antibodies eventually attacking this filtration barrier. Now that's when we actually started at last year's ASN, so I'm telling you, a journey from just less than, or just a year, or a little bit longer than a year, because we were trying to set up these assays before. But so we really tried to set up a team, a collaborative team, studying and trying to detect anti-nephrin antibodies. I would like to mention our lead authors, Felicitas Hengel, Nicola Thomas, of a proteomics team in Hamburg, nephropathology team from Hamburg, but also Astrid Weins from Boston, and then mainly European cohorts like Julien, Pierre Ronco, our Hamburg cohort, the Rome cohort, the Bari cohort, which very quickly opened up their very long and old-standing registries, which were necessary to study really the role of these anti-nephrin antibodies. Now, the problem was at this time that there was no robust detection method. So there was a lot of controversy at the last ASN, actually, because nobody believed the data, nobody could measure the titers. Maybe these antibodies exist, maybe they don't, assays weren't robust, results were conflicting, and the reason is that we are eventually, seeing a novel disease type with low-level circulating antibody titers that rather, and we will talk about the mechanism later, interfere kind of a functional disease than a complement-mediated disease. So we experienced the same thing when we started. So we couldn't just Western blot with patient serum against recombinant nephrin. It wasn't detected, and if you use conventional ELISAs, there was not a robust signal. Even if you use enhanced ELISAs, there was not a robust signal. So we totally confirmed the confusion in the field. And then the team moved on, and with a simple but eventually clever idea at the time, going back to immunoprecipitation. So using recombinant nephrin as a bait to capture all circulating antibodies eventually existing in the patient's serum, pulling then, with our bait, pulling with the antibodies the nephrin down, and then plotting for nephrin, seeing how much nephrin was captured. And then many little details were important, which we can discuss also later in our faculty discussions. What nephrin to use, which cell line to use, glycoside. I mean, many things play a role. So it required some understanding of nephrin biology. So, and then in the next step, and by this, actually, we were now able, with this immunoprecipitation, to get very clear results. Yes, no, binary results. Out of a sudden, all the confusion cleared, and we could see very clear results. Now, in the next step, we used these immunoprecipitates, and we used this streptecnephrine, which we now could then, if used as a bait, precipitated by the patient antibodies, we could put it on the plate, and then on top of this use an ELISA. So by this two-step IP ELISA hybrid assay, which we also patented, which I should disclose, we were now able to quantitatively measure antibody titers. So now the game was opened for serious investigations. This is the largest cohort was coming out of Hamburg, and it shows the other European cohorts that we used. And these are the results, epidemiological results. So in the adult space, we detected these antibodies in 44% of minimal change patients, 9% of primary FSGS patients, and basically it was absent in all other conditions and controls. Interesting, to get to the real prevalence of this disease was looking at the subcohort, minimal change disease, active, nephrotic, not on immunosuppressants yet. Right? So this is the active group. And there, the prevalence was as high as 69, almost 70%. Identifying it as the main association. Now then, when we moved on to children, same thing. Overall, we saw it in around 50%. But again, the interesting group was idiopathic nephrotic syndrome. We didn't have biopsies from these kids. It was the largest European pediatric cohort of idiopathic nephrotic syndrome. And if you look at the patients that were nephrotic, active, not on immunosuppressants, the incidence was as high as 90%. So next question was, okay, so we see a high level of association with these antibodies to the disease. Next question was, of course, does this correlate with anything? So for that reason, you can see here kind of like a traffic-like system. This shows positivity for antibodies in the single patient, 18 patients, and the lower panel shows the extent of proteinuria. And as you can envision here, there was a very high level of correlation. Antibodies present, proteinuria present. No, proteinuria present, antibodies present. Antibodies are being resolved, proteinuria being resolved. So there was a very strong correlation suggesting pathogenicity. Just suggesting, this is just still correlation. So let me summarize this first part of this endotyping adventure. So basically, we could show that there is a high prevalence of antinephrin antibodies in minimal change disease in adults and idiopathic nephrotic syndrome in kids. And we saw a clear correlation of these antibodies to the clinical symptom of proteinuria. Next question, and of course, was molecular mechanism. So is there direct pathogenicity, and how can we understand the role of these antinephrin antibodies? For that, we did an active immunization model, immunized mice with mouse nephrin. We got an explosive onset proteinuria. We got antibody titers. We got a minimal change-like phenotype. We could see some IgG staining pattern at the kidney filtration barrier, where we did immunogold against IgG in places where the slit diaphragm was still remain. We could see localizing the immunogold to the slit diaphragm. And when we then did proteomics analysis, we could see, very interestingly, a massive increase of nephrin tyrosine phosphorylation in response to these antibodies, also again here by precipitating tyrosine phosphorylated nephrin. And on the proteomic level, we saw protein shift of slit diaphragm associated molecules, including CD2AP, which has been identified and cloned here at WashU, and other slit diaphragm associated molecules. We saw strong changes in podocyte adhesion molecular machinery and also actin binding, indicating that the signaling pattern is eventually changing in response to antibody binding to nephrin. Now, these are yet unpublished data because the real Koch-Lesch's postulate and proof of concept is if you do the passive transfer. Just use the patient antibodies and bring it to an animal model. Again, not an easy experiment. Because, first of all, and again, it was led by Felicia Sengel, Nicola Thomas. So first of all, we really needed to look for the cross-reactivity of human antinephrin antibodies across all species. And it turned out, the cross-reactivity to murine or rat is not that good. And it turned out to be best in rabbit. So we used rabbits for this purpose. And then if we did so, it induced proteinuria within 3 to 5 days. First of all, this is from a patient that had a massive life-threatening nephrotic syndrome, one of the rare cases where we even did plasmapheresis, and then we treated the patient. Finally, we took him up, and you can see in red the antibody titers going down as the proteinuria in parallel, so treatment was effective, and at this stage, as we collected the patient's serum, this is what we injected into the animal and received proteinuria and food process effacement. So as a passive proof of concept, here again seeing the food process effacement, the IgG deposition, and the minimal change-like appearance. So how is this now all puzzling together? So obviously what is happening is antibodies are binding to this transmembrane receptor nephrin, key component of the slit diaphragm. Now when we look for complement, we don't see any complement activity or deposition. So we believe it's not an FC receptor-driven biology. It's rather complement independent. When this antibody binds in the filtration slit, with all the 180 liter of primary urine fluid passing, probably also washing all eventual complement away, we see then a phosphorylation of tyrosines and nephrin, upper-nephrin binding. And we know what this is being inducing because these are pathways, and I will show this in the next slide, we've been studying for a long time, also at WashU. But this leads to nephrin tyrosine phosphorylation leads to signaling programs leading to food process retraction. Let me show this here, because these are programs that we have been starting working together, even when I was in Freiburg, together with Andre, when we were showing that this nephrin signaling induces AKT and survival tone and many other signaling pathways. And then later we could show cytoskeletal pathways. Here in WashU, we could again show the importance of a non-receptor tyrosine kinase FUN being recruited to a complex, contributing to the stability of a complex. So everything we have been investigating almost 20 years back. And we did these studies to better understand genetic mutations causing proteinuria in nephrin. But then, at the end of these talks, also here in the signaling seminar, they ask us, what are the upstream signals? What is inducing nephrin signaling? And remember, we didn't have a good answer. You were saying, yeah, maybe, whatever, and pressure might transduce to change signaling, but we didn't have a real, clear, convincing answer. Coming back today, this is the clear answer. It's the most common acquired proteinuric disease in children, where these antibodies bind nephrin, inducing nephrin signaling, leading to signaling programs that in turn result in food process effacement and an opening of the filtration barrier. So this is 20 years old again. That's what we entertained, but we didn't know what the potential upstream signal could be. And I think now this changed. Something that Andre and myself discussed, like in old times, having a coffee and discussing science. And Andre brought up this paper, how we eventually can better understand the signaling. And this is a conceptual paper which conceptualizes condensates. What is a condensate? A condensate is like a cellular organelle, while it's not an organelle being separated by a membrane, but it's an organelle being defined by the density of molecules brought together around receptors. You could maybe call it even a signalosome. And in this paper, they describe, because it's so interesting, all the pathways that we have been describing more than 20 years back, as one of these condensates. So there is really almost a phase separation bringing together these signaling molecules around the receptor. And now, of course, we can go, using such a model will be very interesting to see how this condensate is behaving in the presence and absence of these antibodies. Number one. I think number two as an outlook, it reaches back to work which we did together with Jeff when we tried to better understand the molecular makeup of these molecules. And this was the first study to use cryo-EM on native kidney tissue. And when we solved the molecular architecture, the multi-layered structure of these nephrin molecules. And we also found that these, by computational prediction, that these molecules are rather flexible in their IgG domain, so they are not the actual barrier to restricting albumin. It's not. That's a different function. It's a signaling function. But it's not a mechanical function holding back albumin. But what happens if these signaling properties are being impacted? Food processes are retracting, and this directly impacts the basement membrane. Obviously, if there's less pressure and force from these food processes on the basement membrane, the GBM texture is changing. It's widening up, and it's getting more permeable towards albumin. For that reason, we are becoming a bit more GBM-centric in the future. And this is the mother institution of GBM studies. It started really by Jeff works with Joshua Sainz at the time at neurobiology with some of the landmark paper identifying some of the components, including laminins that build up the glomerular basement membrane. And also here, Jeff has been leading the field of GBM biology now for exactly, I keep bringing up these anniversaries, but it's fine. I don't know, it's for whatever reason, but this is your anniversary again. 30 years of successful GBM research, 30 years as of today. I have no idea why everything falls together in this place, but it's It's really strange. It is what it is. I can't do anything about it. And Jeff is now using this, Hanis Suleiman and others, highly resolved techniques and running perspective projects on highly and using expansion microscopy and super resolved microscopy to better understand the dynamics and the composition of the basement membrane. Now let me summarize this mechanical part. So it seems that we identified, eventually even endotyped a novel disease entity. It's antibody mediated, but it's not FC complement driven biology. It's rather functional interference with a receptor leading to change signaling programs inducing the disease. So now we have, it's clinical grand rounds. So what are the clinical consequences of this finding as of today? So this shows one patient. This was one of the patients that we published, where we use our quantitative assay. It's a frequently relapsing minimal change patient. You know all these patients. You go through different immunosuppressants, and now everything makes sense. Retrospectively, plaque is proteinuria, read as antibody levels. You see this correlation and the relapse of immunological relapse in parallel with clinical relapse. And then finally, this patient received rituximab, and you have an immunological resolvement as well as a clinical. Now, also in our first publication, we also used this to prospectively treat patients. These are two patients, frequent relapsing minimal change in FSGS patient, and then in one of their relapse episodes, high levels, proteinuria, and we decided to directly apply rituximab to target B cells, and we see an immunological remission as well as a clinical remission. And this is a very current case, not published yet or not submitted, where we were contacted from a team in Manchester with a patient that suffered from severe nephrotic syndrome and thromboembolism in cerebral sinus venous system, lung, gastrointestinal tract, so massive thromboembolisms due to the massive nephrotic syndrome, heavily anticoagulated, so biopsy couldn't be performed. We asked him to send us the serum. We measured sky-high antibody titers based on this results. The patient received rituximab and went into clinical and immunobiological remission. So let me summarize this part. So basically, I think we are coming really to the idea that we can use these antibodies that are the pathogenetic concept also for guidance and monitoring of disease activity. And this brings me to the next chapter, which is the clinical spectrum. What more can we learn about the clinical spectrum of these antibodies? Now, key question was, our course that I've been showing has been steroid sensitive, idiopathic nephrotic syndrome, minimal change, patients that respond very well to immunosuppressants. How about genetic cases? Many genetic cases for unknown reasons are even receiving immunosuppressants. And so what can we learn from steroid-resistant and genetic cases? And the largest cohort in Europe is the Podonet cohort run by Franz Schäfer, again a paper under review. And Franz provided us with this 165 children, very well genetically mapped, and then he distinguished these steroid-resistant and genetic cases and non-genetic cases. And the non-genetic cases, he then again looks whether they are multi-drug resistant, don't respond to anything, or intensified immunosuppressants, some patients that respond at some time to whatever combination of immunosuppressants. Now, if you look at the data here, very clear, we just detected the antibodies in one genetic case. The key mess, and this was the Schinke syndrome, which does cause autoimmunity and lupus-like and antibodies at all things, right? So basically, I think the key message here is in genetic cases, we don't find these antibodies. So there's no good reason to treat genetic cases with immunosuppressants. Oh, interesting. So after getting stuck in a power outage in the elevator in the chase, this is now another interesting thing. This computer is dead as well. Let me check the time. Or are we running out of time? No, we're good in time. No, I tell you a story. So basically, looking at these now steroid-resistant cases, the outcome is we don't find the antibody in genetic cases. And then if you go to the non-genetic cases, we don't find them in multi-drug resistant. So people and kids who don't respond to immunosuppressants. This was quick. Thank you. This was quicker resolved than getting stuck in an elevator at the Chase and with a power outage. Second panic moment. You know, it's always good. Thank you. So, but the interesting thing was then looking at multi-drug versus intensified immunosuppression. So in the intensified immunosuppression group, so patients that somewhat at some course of immunosuppressive therapies respond, there we found it in 18%. So I think take-home message here is that these antibodies are mainly found in immunoresponsive patients, makes eventually sense. And the less patients are responding to immunosuppressions, retrospectively, the less likely we find the antibodies. Now, how can we translate all this to future new therapies? I was talking, of course, of refinement and repurposing, which we all do. You know, refinement is when do we use rituximab. Repurposing would be using, if people don't respond to rituximab, using anti-CD38 and so on. So there's a lot of things now to do with B-cell-driven diseases. But I would like to entertain one more idea, going along with the endotyping, bringing the therapy directly to the kidney. The kidneys are the most spectacular and complex organs in the human body, so we would like to treat the kidneys exclusively. How could this be envisioned? And I would like to suggest an gene targeting via AAVs, adenovirus-associated viruses, and they have been FDA approved throughout many organs, not the kidney yet. They have a broad tissue tropism, long-lasting efficacy, low immune response, non-replicative, so they are potentially good tools for gene transfer. Now, not again. Oh, no, good. Okay, now, basically what we did, also something that we patented, we developed a selection method to, based on AAV libraries with 10 to the 8 different peptides displaying on the surface, and then basically we inject them into animal models, we harvest all organs, we look for the enriched AAVs, and then we repeat these cycles 4, 5, 6 times, and then we have a mathematical algorithm telling us, you know, that we look just for enriched in the kidney, but not in other organs, and by doing so, we came up with one amazingly, in the first ever kidney-specific AAV. It's amazingly enriched in the kidney. It targets specifically glomeruli. Of course, we hoped it would be podocytes, which was a little bit naive because these things are not crossing a healthy, intact filtration barrier. It co-labeled with glomerular endothelium. Remember, glomerular endothelium is very specific. It cannot be compared to any other endothelium in the body, large fenestrae, large glycocalyx. So it has its own biology. And for that reason, it seems that it can be targeted specifically. Now, we used this as a concept, proof of concept study. Remember when we talk about autoantibody-mediated podocytopathies and diseases. So why don't we prevent these autoantibodies from reaching their target, the podocyte? And for that reason, we used IDAS, this is short bacterial protease that cleaves all immunoglobulins, and we delivered the IDAS to IDAS sequence to glomerular endothelial cells so that they express IDAS. And when we did this experiment, we could 100% prevent even the most aggressive antibody-mediated, even Emil Onano's NTS glomerular disease, which he thought could never be prevented, but it can now be prevented by using AAVs. And there is no antibody deposition, there's no whatsoever complement activation, so they are 100% protected. It's a proof of concept. When we are moving on to pigs, we think this can be a very useful tool for xenotransplants, actually. Last but not least, you know, if we want to move forward our field, it's only done together, it's only done in networks, it's only done synergistically. It's only done with the wash-you spirit of team medicine. And for that reason, we recently founded a new society, the International Society of Glomerular Disease, really bringing all glomerular disease experts together. Since we now move on with new disease entities, we have 64 trials into phase 2, 3. So a lot of new glomerular medicine is coming, and for that reason, we needed a society focusing from glomerular disease to glomerular medicine to glomerular health. And this new society is now rolling out in 90 countries across the globe, most prominently in the United States. That's also where we are hosted as a nonprofit organization. We have 1,200 members. We are trying to create roadmaps for glomerular diseases as a nephrology subspecialty for glomerular disease fellowship. I hope you have one here. And for building up glomerular disease centers, but also to identify clinical trial platforms, to generate online tools, patient-to-trial matching tools, and so on and so forth. And I just would like to entertain one most recent thing is, so we identified that there are no surrogate endpoints for FSGS. It's probably the glomerular disease with the largest unmet need. And under the lead of our society, together with NEFCure, the Kidney Health Initiatives, and the National Kidney Foundation, we built a consortium, an altruistic consortium, in 28 registries across the globe, most coming from this country, opened to allow a retrospective study to see how proteinuria associates with hard endpoints, and you really see the outcome that kidney failure and kidney-free survival really depends on the level of proteinuria and how patients respond initially, and whether proteinuria is maintained or whether proteinuria can be resolved. And it looks like, and we're doing this together with the FDA, we introduced this data at this ASN meetings, and we are kind of optimistic that the FDA by the end of this year might approve proteinuria as an accelerated surrogate endpoint, because GFR slopes didn't work for this heterogeneous disease. And if this comes true, a lot of pharmaceutical companies are already lining up. It will be a game changer. But again, this is nothing that one team, one university can achieve by themselves. It really requires large-scale thinking in terms of collaboration. And that's why we needed this society, and that's why we need to interlink also all our universities. So let me summarize. So kidney diseases are a major threat. They're an epidemiological catastrophe. But if you look closer, the main unmet need are rare kidney diseases, accounting for 30% of renal failure, and we don't have good answers yet. For that reason, we worked on, we suggested an endotyping concept, and we worked on a new entity, the anti-nephrin autoantibody podocytopathy, which accounts for a major fraction of minimal change and also idiopathic nephrotic syndrome. And we found that this is a low antibody titer disease, probably inducing disease via signaling programs. The less people respond to immunosuppressants, the less likely they have these antibodies, and we can use now insights, and we are gaining now, of course, prospective insights and prospective studies to better use these biomarkers and pathogenicity markers to guide our therapeutic decisions. I also kind of introduced novel concepts, how we eventually can bring therapies directly to the kidney, alongside the better molecular and single-cell resolved understanding, as we see it here at WashU. And this basically sums up the concept of endotyping. So let's think in kidney diseases in separate disease factors from disease. The disease itself is being originated in the tissue. That's what we call the endotype if we identify the pathological drivers. This can be translated to a new scale of matrix of diagnosis, the DCT system. This directly translates to better refinement, repurposing of drugs, and also developing eventually novel drug pipelines. And if you project our new example, then you would see this, the CKD disease factor is autoimmunity, autoantibodies. This would be the disease factor. It's not the disease, it's just the disease factor, right? Then the circulating factor is the antinephrin antibody, and that's now coming to a name. And this basically, and the tissue response is the podocytopathy in response to this change signaling programs. So following this path that I suggested at the beginning, this comes, and let's do this today. let's reclassify today minimal change disease and idiopathic nephrotic syndrome as, if anti-nephrin antibodies are being found, as an anti-nephrin antibody podocytopathy. So let's start, as of today, naming the endotype instead of a histotype. So if you're interested in glomerular diseases, you might join the Hamburg podocyte meeting next year. I guess quite a few people from here will be coming. You can join also the society. And the last picture should be dedicated to Eduardo Slatopolsky, and from him being an inspiration for us way beyond his life by bringing back complex understanding back to patients and doing this with the same enthusiasm and hopefully endurance as he did for his whole life. Thank you very much.
Thank you so much for that inspirational tour de force and very appropriate Slatopolsky lecture. We're now open for questions. I might lead off with the first, and that is, and I'm sure you've thought of this, upstream, how do the antinephrin autoantibodies arise? There is, of course, in minimal change, or I take it back, antinephrin podocytopathy, clinical syndrome, a correlation sometimes to a preceding viral illness. Where is the nephrin-like antigen being exposed?
No, it's a great question. I need to have a few more coffees with Andre afterwards to discuss this as well. No, we don't know yet. Of course, we are looking into molecular mimicry. There's a clear correlation of infections, particularly in kids, to these antibodies being found. So for that reason, we are very closely looking whether there is somehow a direct or hidden molecular mimicry. And beyond that, we also would like to better understand the B-cell biology. There might be, I tell you a secret, there might be, in the few recurrent FSGS cases that are antinephrin antibody positive, these very aggressive, not minimal change histotype-like, but FSGS histotype-like diseases, we find these antibodies to bind to different epitopes in nephrin than the regular benign. So, we need to understand more, and also where these B cells and plasma cells are eventually be sitting. So I think it's not originating in the kidney, the disease is originating somewhere else. That's why we need to go to these places to better understand the origins of the disease.
Leslie.
So congratulations, really a great example of the combination of team science and molecular biology techniques to change our understanding of this anti-nephrin podocytopathy. My question actually relates to the AAV work that you're doing. I think that also has really incredible potential for research and for treatment. Have you looked at all in your work with that on the proximal tubule cells of target?
Yeah. So I'm asked to repeat the question. So the question is regarding the AAV work, are there any tubular-specific AAVs? We haven't found in our selection process anyone yet because they're not being filtered, so it must come from the basolateral side. There are quite a few companies that are looking because they also want to target PKD and other genetic diseases. We haven't found one, but work is ongoing to detect tubular-specific and even tubular compartment-specific. We found one, interestingly enough, there is an affinity towards the macula densa. So we found we found even a more specific AAV, highly enriching in the macula densa, which we could think of using to manipulate blood pressure and other interesting things.
Maggie.
Yeah, so the question is whether there could be antibodies directed against other slit diaphragm components. Great question. There was a very nice ASN abstract poster showing an antibodies against CRAMS2, the polarity component, also being enriched in the slit diaphragm. We've been looking for NEF1 and others. We haven't found yet, but now since we have a population which is antinephrin negative and proteinuric, this is our population where we now use strategic experimental setups to look systematically for new and additional autoantibodies as we speak.
I'm sure Toby will take questions here at the front, but once again, thank you for a terrific Grand Rounds.
Thank you.